Hey, that was a wonderful way to introduce the next session as well, which is another innovative project that's out of the University of Toronto and brings together multiple stakeholders that really want to advance the field. And, we have this project that brings together MyTax, a non-for-profit organization, industry and academic lab to develop mitochondrial gene risks for patients with bipolar disorder. So in the next segment, I'd like to welcome Jaehyoung Choi, Joseph Geraci, and Kate Murray, who will all be talking about this exciting project. Jay is a third-year PhD student in Dr. Anna Andreas's lab in the Department of Pharm and Toxicology. He focuses on bioinformatics analysis of mitochondria-related molecular alterations in complex neuropsychiatric disorders. And he's currently collaborating with Neuroscene to develop a novel pipeline for the quantification of mitochondrial dysfunction risk in neurodegenerative disease through artificial intelligence. Dr. Joe Geraci is a mathematician, medical scientist, and a machine learning scientist. He has spent the last decade creating novel methods that enable machine intelligent methods to perform better with complex patient population data on oncology, neuroscience, and aging. He is the founder of Netramar Corp, which was recently acquired and holds 2 adjunct faculty positions at Queen's University, Molecular Medicine, and August University in the Center for Biotechnology and Genomics Medicine. His particular interest is in the application of biologically inspired explainable AI methods in order to create a new taxonomy of disease with clearly defined etiologies. And Kate Murray, who's a stranger to Mito2I and to our symposium and conferences, is the current co-CEO and president of Mito Canada Foundation. Kate has an extensive not-for-profit experience working for small charities and foundations, founding her own children's charity, Robbie's Rainbow, with her son. And previous to the not-for-profit life, Kate's career began in the food and beverage industry, working for one of the largest brands in the world, Coca-Cola. As one of just 3 women to hold a distribution management position within Coca-Cola Canada, Kate turned one of the country's poorest performing locations into one of the top 5 in North America. So I'd like to welcome Jay to introduce the project, Joe to give us a little snapshot of its applications, and Kate to talk about the patient engagement pieces for this.
Hi everyone, and Sonia, thank you very much for your introduction. My name is Jay, and I am a PhD student in the Anna Andreas lab at the University of Toronto. I'm a pharmacologist and a data scientist in training. And I am very delighted to present our current collaborative project between Mito Canada, Neuroscene, and our lab, focusing on nuclear mitochondrial genes in neuropsychiatric and neurodegenerative diseases. So things that you've probably already heard about. So mitochondria are very important organelles in the human brain, especially because the human brain uses about 25% of total bodily glucose, about 20% of bodily oxygen that we're supplied with, where glucose and ketone bodies are very rapidly metabolized by the mitochondria. And the cells of this energy expensive organ are supported by dense and active networks of mitochondria to support their function and maintenance. As such, mitochondrial dysfunction may lead to critical problems in the brain, and it has been implicated in various psychiatric disorders and neurodegenerative diseases. So as with a lot of the panelists and audience here, but I can probably spend an entire day about cumulative evidence of mitochondrial dysfunction in many brain psychiatric disorders at the biochemical and biological levels. So I just pulled up a few examples, recent examples of mitochondrial dysfunction in different psychiatric and neurodegenerative diseases. So for example, there has been recently found a decreased mitochondrial copy number in periphery of patients with ADHD. In the middle there, that would be postmortem imaging and mitochondrial structural abnormalities in schizophrenia, bipolar disorder, decreased brain pH, and deficient OXPHOS components in bipolar disorder. And furthermore, the psychiatric conditions present in early to midlife, which is the regular onset, they pose a very high risk of developing forms of geriatric neurodegeneration in late life, in which mitochondrial dysfunction is one of the leading hypotheses in the pathology of Alzheimer's disease and Parkinson's disease. So mitochondrial genetics. Mitochondrial genetics, it's super complex because it's in dual genomic control. So there is the nuclear encoded mitochondrial DNA, which composes more than 97% of proteins that act in or on mitochondria. And there has been few hints from large-scale genome-wide association studies about genetic risks, but with no significant pathway hits specifically on mitochondria, there is still lack of knowledge on how mitochondrial genetics may affect the risk of disease development. So this is just an example. This work by Dr. Vanessa Gonzalez was the first to examine specifically the nuclear encoded mitochondrial genes association with schizophrenia, which gave some new insights into mitochondrial genetics. And then our project has an identical framework to focus specifically on mitochondrial genes with diseases. Okay, so here's like my train of thought. Okay, so we know that mitochondrial dysfunction can lead to psychiatric phenotypes, as about 30 to 40% of patients with primary mitochondrial diseases, which are genetic diseases, have comorbid mood or psychotic symptoms. But there are still heterogeneous findings on detection of mitochondrial dysfunction in psychiatric disorders. We still don't know whether mitochondrial dysfunction is involved in primary pathogenesis for these patients or is a secondary hallmark which we can sometimes observe, or if mitochondrial dysfunction affects all or some of the patients with this brain disease. And the truth is that using standard statistical methods based on hypothesis-based testing, it's very, very difficult, or it may not even be possible to work through this heterogeneity with single disease labels. So we'll implement different machine learning techniques to identify patient subgroups which altered mitochondrial patterns in gene expression and genetics kind of signify those subpopulations and identify clusters with shared molecular profiles and investigate cross-disorder risk associated with these patterns, with these altered patterns in mitochondrial genes. So, oops. Sorry, I'm having a little bit of trouble. Hence, we'll be running through a parallel pipeline of supervised machine learning, unsupervised machine learning, and ResNet-based convolutional network, and Neuroscene or Netramark's exciting Netra AI to examine a mitochondrial risk score to characterize mitochondrial dysfunction linked to risk that is associated with cross-disease pathology. So there we go. So here's a very, very short snapshot of our exploratory analysis, where we fed the model with only mitochondrial gene transcripts, which is only a 5% of the entire transcriptome. And this model is able to perform adequately well, telling whether a sample belongs to a case or control in bipolar disorder data set. And we start to examine these interesting clusters sharing similar mitochondrial gene patterns with high patient density. So this is an example. Well, this is a published collaborative work from last year with Netramark and Dr. Joseph Geraci, which analyzed postmortem gene expression profiles from patients with bipolar disorder. So this augmented intelligence platform invented by Netramark identifies patient subpopulation and important genes that segregate different patient and control groups together. And Dr. Geraci will walk through this technology with you shortly. So The main goal of our projects is first to identify mitochondrial gene patterns associated with disease, with evidence of mitochondrial dysfunction, identify the subgroups of population with possible mitochondrial alterations, and most importantly, translate this risk or the knowledge that we've identified to the broader patient-centered community using our individual-centered approach using machine learning techniques. And I'll now pass the mic to Dr. Jirachi to now explain how he shatters the disease heterogeneity. Thank you.